Hello guys, so let us discuss the general introduction of the microbiology regarding the scientists and all and what they have invented or discovered, right? So this is very important, okay? So let us start with the famous scientist, all of you know, who is also called as father of your microscopy, right? So he is none other than Sir Antony van Leeuwenhoek. Antony van Leeuwenhoek. Now, who is this Antony van Leeuwenhoek? What are the important things you can know about this scientist Antony van Leeuwenhoek? So basically, he is considered as the father of microscopy, right? So this is one of the very important MCQ. So who is considered as the father of microscopy? Yes, father of microscopy is Antony van Leeuwenhoek, because he is the one who invented the microscope. He is the one who invented what? Invented your microscope. And once after inventing microscope, what he'll do, obviously he'll try to find some microorganisms, right? So he invented the microscope and he found microorganisms right beneath the microscope, and he named these microorganisms as little animalcules, right? So he named these little small microorganisms as little animalcules. Okay, so what did he tell? He also told he named the microorganisms as little animalcules. Right? So these are the important things which are pretty much enough for you for the exams regarding Antony van Leeuwenhoek. Now next we shall be switching on to Sir Louis Pasteur. Now very important things to know about Louis Pasteur is that, so once upon a time Louis Pasteur, what did he did is that he has taken his car, okay, right, and you know this is a car from Germany. He has taken his German car and what did he do? He completely washed that car. Washed with what? Obviously you'll washed with some liquid, right? He completely washed that car and went on a trip. Clear? Now what you need to understand from this? What you need to understand is, in fact what I have written is nothing but called as your mnemonic, right? So what did he give us today? What Louis Pasteur has given us today? So the important things that Louis Pasteur has given us today is, German stands for germ theory. Germ theory of the disease. So Louis Pasteur has given a germ theory of the disease. Now what is germ theory? Simple thing guys, germ theory means he told that, look, there will be some microorganisms which will invade your body and cause disease, and you can see these microorganisms only under a microscope. With your naked eye you cannot see it. This is what he told. Okay, in the form of a definition he told it, that's it. Okay, what is a germ theory exactly? He told that there will be some microorganisms, there are some microorganisms which invade the human body and cause a disease. And these microorganisms, where can I find? I cannot find with my naked eye, right? I cannot look them like this, only I can look them under a microscope. That is what is called as the germ theory of the disease. Okay, and second important thing, after the germ theory, what is the thing is that he is the guy who coined the term vaccine. He is the guy who coined the term vaccine. And in fact, he discovered vaccines of 3 important diseases, you know. He discovered vaccines of 3 important diseases. What are those 3 important diseases? C stands for cholera. C stands for cholera, and A stands for anthrax. A stands for anthrax, and R stands for rabies. R stands for rabies. So these are the three important things. CAR, CAR, cholera, anthrax and rabies. Fine. What is the third important thing? Third important thing is that he also gave us techniques how to sterilize. Sterilization is very important nowadays, because you are using many surgical instruments, you are using, you use the operation theater, you use the materials for the operation theater, you use the cotton pads, gauze pieces, right? And for all of this, you cannot simply use them like that. You have to sterilize them before using, right? So you have to know the techniques of sterilization. Sterilization is very important to kill the microorganisms, isn't it? So he wash. What do you mean by wash? Wash means you are washing away all the microorganisms. It means you are sterilizing. So he gave us methods of sterilization. Not only that, he washed with what? Liquid. So he also has given us a media for the microorganisms culture, that is called as a liquid media. So he has a German car who washed with the liquid. Simple thing, right? Apart from this, what is the fifth and sixth important thing is that? Look here very carefully, guys. So Louis Pasteur, right? So P stands for two things. P for Pasteur and P for principle. So he has given a technique called as pasteurization technique. Let me write it and then explain you. He has given a technique that is called as pasteurization technique. You know what is pasteurization technique? Pasteurization technique means simply, mostly pasteurization technique is used for milk. Okay, we call it as pasteurization of milk. You heard it, right? So what is pasteurization of milk is that simply killing the microorganisms in the milk is called as pasteurization of milk. Okay, you will have some temperatures. Only at those temperatures you can kill those microorganisms. Okay, I'll mention them. I'll mention them in a second. So, but before that, just understand what is pasteurization. That's it. And what did I tell you? P. Another P, right? P for pasteurization. P for principle of fermentation. Principle of fermentation. I hope all of you know what is fermentation, right? Fermentation is simply converting sugar into an acid or alcohol or a gas. Okay, if you are converting sugar into acid, alcohol or gas, that is called fermentation. But you have to convert it in the absence of oxygen. Keep that thing in mind. In the absence of oxygen, if you are able to convert sugar into acid or alcohol or gas, this process is called fermentation, and you are called as a fermenter because you are able to do it, right? So if the bacteria is able to do it, this is called as, those bacteria are called as fermenters. Okay, so what bacteria are going to do, we shall be discussing that. But these are the basic things you need to know. What are the basic things? The basic things are, simple thing guys, once again, Louis Pasteur. Okay, so he has got a German car who every time used to wash with the liquid. Okay, so German in the sense germ theory of the disease, I explained you what is that. Second thing is that he coined the term vaccine. In fact, he discovered 3 vaccines. One is cholera, anthrax and rabies. Okay, washed in the sense sterilization, wash out. Next in the sense liquid, in the sense liquid media. And P stands for 2 letters, P for pasteurization, P for principle. Pasteurization means pasteurization of milk. Principle means principle of fermentation. Okay, so these are the important things you need to know. So after giving all these 6 things for us to study in microbiology, don't you think he is an important person? Yeah, see, Antony Leeuwenhoek has given us microscope, so you called him as father of microscopy. Now Louis Pasteur has given you all these 6 things, then even you have to make him a father, right? So can I call him as father of microbiology? Yeah, so let me write it as the seventh point, that he is considered as father of father of microbiology. Okay, keep these things in mind. So we were discussing that there is a technique called, I will go into detail of these techniques in the next session, but I'm just giving you a brief idea. Okay, so I've told you that there is a technique called as pasteurization of milk, right? So in this technique of pasteurization of milk, what you need to understand is that, see, you take a milk, either it is a packed milk or a milk which is not packed or non-packed milk. What do you do is that you heat it to some degrees temperature. When you heat it, what will happen? The microorganisms will go away. So when it will go away, it means you are increasing the life of the milk, you are increasing the shelf life of the milk, isn't it? By removing all the microorganisms, you are increasing the shelf life of the milk, and that is called pasteurization. So what are the three simple steps, guys, in pasteurization? What did I tell you? Yes, either you take a packed milk or you take a non-packed milk, packed or non-packed milk. Okay, what you do is that you heat it. Okay, so you will provide some heat. Now when you heat it, what will happen is that the microorganisms get eliminated. So there is gonna be elimination. Elimination of what? Elimination of your microorganisms. Now because of this elimination of microorganisms, indirectly what you are gonna do? You are increasing the shelf life. So this is the important thing to keep in mind. This is the important thing that you have to keep in mind. So these are the three simple steps which you're gonna do. Now how you will do it? Okay, you're telling me that I will heat it. Okay, so you are telling me that you're gonna heat it. Now I am asking you, how will you heat it? And again I'm telling you, I use two methods to heat it. Now this is one method, and here I am writing down another method. Okay, now what is the first method? What is the first method is that, simple thing, guys. In the first method, I will heat the milk at 63 degrees centigrade. Okay, let us say I am heating the milk for 63 degrees centigrade for about 30 minutes. 30 minutes. Okay. In other method, what I will do is that I will heat the milk at, let us say, 72 degrees centigrade. 72 degrees centigrade. Now you tell me, I have increased the temperature. If I am increasing the temperature, I have to decrease the time, because let us say I am heating something at 10 degrees for 10 minutes, that's fine. But I am heating something for 100 degrees, right, at 100 degrees for again that same 10 minutes, then what will happen? The milk will completely evaporate, isn't it? So what you have to do is that when you are increasing the temperature, you have to decrease the time. You're getting it? So 63 degrees for 30 minutes, that's fine. But if I have increased the temperature to 72 degrees and if I am still keeping it 30 minutes, then what will happen? All the milk will undergo lysis and it will evaporate. So I should not do that. I have to decrease the time. So 72 degrees, keep this thing in mind. Everywhere keep this thing in mind. 72 degrees for about 20 minutes. Not 20 minutes, for 20 seconds. 72 degrees for 20 seconds. Okay. Now look here. If I am heating the milk at 63 degrees for about 10 minutes, then all the microorganisms are going to die except one, that is called as Coxiella burnetii. Okay. So what am I going to write? All microorganisms All microorganisms are killed except Coxiella. Coxiella burnetii. Okay, Coxiella burnetii. Except this, all your microorganisms are killed. And this method you call it as Holder method. This method you call it as Holder method. Holder method. Okay, now when I'm heating at 72 degrees for about 20 seconds, what is going to happen is that all microorganisms are killed, all of them, including this Coxiella, everything is killed. Okay, so all organisms are, don't think it as organs, it is organisms. All organisms are killed without any exceptions. All organisms are killed. Okay, so this method you call it as flash method. For 20 seconds, within 20 seconds, like a flash, suddenly all of them are gone. This is called as flash method. This method is called as flash method. Clear? Now this is all you need to know about pasteurization, right? You are not going to become a microbiologist in the future, right? So to know everything in detail, so this will be enough for your exams. Okay, this will be enough for your exams. So for your MBBS prof exams, so pasteurization of milk, this is the important things you need to know. Clear? Okay, next important thing is that there is another method to I told you sterilization, right? You are gonna sterilize it. So how you gonna sterilize? He also gave us techniques of sterilization. How you gonna sterilize is that by two ways, guys. Okay, by two ways. One is hot air oven. Hot air oven. Previously what I have discussed is pasteurization of milk, killing the microorganisms in the milk. Now what I am doing is, for example, you have performed a surgery, you have used the scalpel, you have used the scissors, you have used, right, artery forceps. Clear? You have used all the trays, the kidney trays and all, everything. Now after that you have to sterilize those instruments. You cannot wash it with the soap, right? You have to sterilize it, because who knows, see, maybe you have operated on a patient who is having HIV. Maybe you have operated on a patient who is having hepatitis, HbsAg positive. So in those patients, after operating, or any kind of patients after operating, you have to clean those instruments, isn't it? Right? You have to properly sterilize it. So for those instruments you have to use higher temperatures or lower temperatures. Obviously you have to use higher temperatures, only then the microorganisms on the instruments will go away. You're getting it? So for that I use a method called as hot air oven. Now in this hot air oven, what will I do? Okay, first of all, let me write down a list of what all things I'm gonna work on. All kinds of lab equipment. On all kinds of lab equipment, jars we have got, petri dishes we have got, pipettes we have got. So all these lab equipment also we sterilize using this hot air oven. Not only that, surgically, scalpels and even blades, scissors and etc. Etc is not an instrument. So these are some of the important things which we basically sterilize. Okay, now you tell me, do we use higher temperatures or very low temperature? Obviously we have to use higher temperatures, higher in the sense 160 degrees centigrade. 160 degrees centigrade for about 2 hours. When you heat them for 160 degrees centigrade for 2 hours, right, then the microorganisms on these scalpels or blades or scissor, whatever is there, they are gone. Actually, guys, you might get it out. Why are you using 2 hours time at 160 degrees centigrade? The reason is, every bacteria, if you look at the surface, okay, let me write it down here. If let us say this is some bacteria, this is a rod shaped bacteria, let us say. On the outer edge of the bacteria, on the surface of the bacteria, you have got a jelly like material. See, whatever I am drawing is a jelly like material. You have got a jelly like sticky material, like a gel, sticky gel. This is called as biofilm. You know what is this called? This is called biofilm. It is with this help of biofilm, these microorganisms are sticking very easily to the metal surfaces. Okay, with the help of these biofilms, these microorganisms will very strongly, it will attach to a metal. Either it might be a scalpel or a scissor or whatever it is, it will attach very strongly to that. So you have to remove that contact and kill it. So you use such higher temperatures for a longer duration. Okay, fine. But anyways, if you think you don't have time, you are working in a, let us say, a government hospital, you have got a lot of instruments to work on to sterilize, and you don't have time to wait 2 hours to sterilize all these things. So what you do is that you go for an alternate machine. Okay, you go for alternate machine. If you think it is too much time taking, if you think it is of too much time taking, you see an alternate machine, and that alternate machine, guys, the temperature which you use in that machine is 121 degrees centigrade for 15 minutes. Now you are really shocked, because previously you used 160, which is more than 121 for 2 hours, but now you have reduced the temperature and reduced the time also. Yeah, how? Because the machine works like that. You know what is the name of the machine? Autoclave. Okay, so in autoclave you basically use a temperature of about 121 degrees centigrade for about 15 minutes. Okay, another important thing is that fermentation. I already spoke about that, right? What did I tell you in fermentation, guys? Yes, what did I tell you? I told you that if you are able to convert a sugar, conversion of a sugar into what? Conversion of a sugar into either an acid, right? Either a gas, or let us say alcohol. Conversion of a sugar into either a acid, gas or alcohol, and in the what? In the absence of oxygen. So let me write it as in the absence of oxygen or without oxygen. Without oxygen, if you are converting it, that is called fermentation. Okay. Now, important thing to know is that all bacteria, whatever bacteria is there, all the bacteria have an ability to convert sugar into an acid. But only few bacteria has an ability to convert sugar into gas. Okay. So what is my statement which I have given? The statement which I have given is that all bacteria produce acid. All bacteria produce acid, whereas only few bacteria produce gas. How will I know? How will I know? Am I going to smell it? No. Simply with your naked eyes you can know it. If that bacteria is producing acid, that entire medium will turn to pink. If it is not turning to pink, it means it has produced gas. Simple example. What I will do is that I will take a medium. In that I will add solution of lactose. Okay, in that I will add a solution of lactose. Now what I will do is that I will add some bacteria. Now after adding some bacteria into that lactose solution, this bacteria, what will it do is that if this bacteria will convert that lactose into acid, then what will happen is that that entire lactose solution, it will turn to pink in color. If that bacteria cannot convert this lactose into acid, then what it will do, it will convert lactose into gas. Then you cannot see any color discoloration, it will be same. Okay, so if this bacteria is able to ferment the lactose, right, conversion of sugar into, I told you, right, into acid or gas is called fermentation. So if this bacteria is able to ferment, you call this bacteria as lactose fermenter. Lactose fermenter. Okay, so the entire medium will turn to which color, guys? The entire medium will turn to pink. If it is not able to ferment, this is called as non-lactose fermenter. Okay, then the entire medium will be pale. Then the entire medium is gonna be pale. If it is pale, you have to think that acid is not released. Okay, let me give you the live example of that, the clear example, that I have taken a container. In that I have added what solution? Maltose. Maltose. I have added maltose. And I have added some bacteria. Okay, I have added some bacteria. Now what this bacteria did is that, okay, I have taken two containers. So in this container maltose, in this container also maltose. I have added a bacteria called as X, I have added a bacteria called as Y. Now here I can see pink color, it means this bacteria is called as maltose fermenter. Here I cannot see pink color, so it means this bacteria is called as non-maltose fermenter. Clear? So there are two bacteria I have added, one is called as maltose fermenter and I hope you understood my explanation, right? So the example of that is Neisseria meningococci. Neisseria meningococci. On the other hand, I have added another bacteria which did not turn into pink color, that is non-maltose fermenter. It is pale only, right? This is called as Neisseria gonococci. Gonococci. Okay, so this is one of the important example which you have to just stick it in your mind. Clear? So the third important person we will be discussing is Robert Koch. Robert Koch. Okay, so who is this Robert Koch? Robert Koch is also considered as father of bacteriology. Father of bacteriology. Okay, so okay, fine. So he is considered as the father of bacteriology. Now what all things did he give us? The first important thing is that, you know, Louis Pasteur has given what media to us? Liquid media, right? So Robert Koch has given us solid media. That's it. So he has given us solid culture media. Solid culture media. Second important thing is that Robert Koch have isolated some organisms. He has isolated, which is a breakthrough, he has isolated some organisms. Okay, now what are the organisms did he isolate is, one is called as cholera, that is Vibrio cholera, and next one is called as anthrax, that is Bacillus anthracis, and the third one is called as TB bacilli, tuberculous bacilli. So CAT, cat. Remember, the first one was vaccines, that is car, this is cat. Simple things. So CA, both of their CA in the vaccines also it is the same, the cholera and the anthrax vaccine. Here it is cholera and the anthracis organisms, right? So he has isolated these three organisms. And third important thing is that someone asked him, can you are telling me that you have isolated the organisms, that's fine, but we all wanted to see how do the organisms look like, what is the shape of the organisms, how the organisms are moving, how the organisms are arranged, what is the size of the organisms, we wanted to see it. Can you do an experiment for that? Yeah. Now he said yes. Now you know what experiment did he do? So he has taken a glass slide, guys. So this is a glass slide, all of you know, right? But this glass slide is completely even, right? On both the sides there are no depressions. But this particular glass slide which he has taken, in the center, exactly in the center here, you will see a small pit. Okay, you will see a small concavity in the glass slide. So exactly in the center you will see a small concavity. Okay, now how does that look is that, so this is the concavity which is gonna see in the glass slide, right? Now what he will do is that, so let me write it down, guys. He has taken a small glass slide. Okay, so this is the glass slide which he has taken. Okay, and what did I tell you? This glass slide has got small concavity in the center. And another thing is that he has taken a small cover slip like this, okay. What did he do is that, on this cover slip he had added a drop, and in that drop of liquid there is bacteria inside that. Okay, now if I am rotating the cover slip, what will happen? This drop, whatever is there, if it is having a very good surface tension, this drop which is containing bacteria, drop of liquid which is containing bacteria, it will hang down like this, but it will not fall off. If it is having a good surface tension, then what will happen? This drop will catch the glass like this, the cover slip like this, so that it hangs like this, isn't it? I hope you can imagine. Now what he will do is that, I mean, he will take this glass slide, whatever is there, that glass slide is having a concavity, right? Exactly on the concavity he is going to place this cover slip, and now the drop will be hanging down. Now through the microscope you can very clearly see the microorganisms. So this is called as hanging drop experiment. Okay, in that drop or bubble, whatever is hanging like this, you can see the microorganisms giggling over there, you can see the microorganisms movement, you can see if they are arranged in a chain or arranged irregularly, are these microorganisms circular or rod shape, you can see all of that. Okay, so what he has done is that he has taken a cover slip, and look here, this is the cover slip like this. Now cover slip has to rest on the glass, so what he has done is that he had added petroleum jelly on both the sides like this, like a suspension. Okay, now what will happen? What will happen is that you can see very beautifully that a drop is hanging, and within this drop you can see small microorganisms like this. Okay, so this is called as hanging drop experiment. What experiment is this? He discovered an experiment, point number 4, that is called as hanging drop. Or method, hanging drop method or hanging drop experiment. Okay, so in this hanging drop, what you can see is that you can tell the shape of the bacteria, you can even tell the size of the bacteria, you can even tell the arrangement of the bacteria, right? And you can even tell the motility of the bacteria. Motility of the bacteria. Motility of the bacteria. So all these things you can tell. And point number 5 is that he has also given us many methods of staining, staining methods. Staining methods. Now I know it will be difficult for you to remember all these things, right? So how do you remember this? I hope Robert Koch hanging drop experiment, right? So the remaining things which you need to remember is like this. SID. I hope all of you know what is SID. SIDS. Okay, simply remember it like SIDS, SIDS. Okay. So S stands for solid culture, I stands for isolating the organisms, D stands for drop, and again S stands for staining methods. Clear? So these are some of the important things which you need to remember. Clear? And now let us discuss the Koch postulates.